So cells with cell wall, they have, you might imagine, a little bit of a challenge when it comes to growing. So if you put on a rigid cell wall and you need to expand your cell as your plant is getting bigger, the new cells that are made kind of inside of the existing cells. So this is the process of cell division in a plant cell that's got kind of a rigid cell wall. So you notice the cell wall isn't changing size at all during this, but we're going from one cell in the beginning to two cells at the end. So the cell membrane gets divided. It's not really illustrated too clearly here where the cell membrane is getting split there. But you can see the new cell wall being laid down, and the cell wall is made as kind of a plate. So it starts off as these little vesicles of building blocks, and they aggregate together, they collect together and make this plate that ends up dividing the top cell from the bottom cell. And you end up with two cells with their own cell walls. So it's sort of adding new cell wall from the inside. I mentioned this is rigid, and the rigidity is not always 100% rigid. Sorry, I'm going over here. So one way that cells can actually expand, of course, you might sometimes need that as a plant cell. If your cell is wrapped around with cellulose, primarily in this direction, then it's basically allowed to expand lengthwise. So plants can control a little bit how their cells grow based on how that cellulose is wrapped on there. So the same strands can just get kind of separated out farther from each other and allow this plant cell to expand that way. So if this was happening as part of cell division, the plant could turn that one cell into two. And in this case, the starting cell is about the size of these two ending cells. So cell wall, not always like 100% stiff. It's not a brick wall. It's more like being bound with twine. And so if you wrap your twine in the right way, you can keep the ability to expand. And then also, just allowing the cell to grow to a certain shape is determined to an extent by where the cellulose strands are located. All right, so I think I used to have a slide on that, and I might not anymore. Cell plates, don't worry about details on here, but just kind of showing how there's some molecules involved. CSC is a cellulose synthesis complex, so getting it in there to make that new cell wall. But before there's a cell wall there, it has to get kind of organized by vesicles and cytoskeleton and so on. As a result of dividing, one thing that happens is the two cells that are produced from division, they have a wall between them, but they might retain, they usually do, retain some connections in their cytoplasm. So there's a cell wall between two neighboring cells, but they have cytoplasm going between them. And not just cytoplasm, but actually, as it's shown here, some bits of organelle. So endoplasmic reticulum can be kind of shared by neighboring cells. So as we'll see when we talk about water transport, the inside of a plant is kind of one shared watery environment, and allowing the cells next to each other to share cytoplasm means that they can communicate, they can share metabolites, and kind of save time on if molecules need to be transported or something like that. Transporting them through the same membrane is a lot easier than like exporting them from a cell and importing them to the next cell. So some illustrations here. This is just from a paper that kind of highlights some of the benefits of the plasmodesmata, these cytoplasm connections. So hormone signals, as I mentioned, so being able to move molecules that are involved in metabolism, metabolites, and then also signals in between cells. So these are quite a few of these are showing how some of these signals get propagated. So growth signals involved here, signals about being injured can be propagated, and so on. And so that's a lot faster by sharing cytoplasm. Sometimes there's a case where the cytoplasm should not be shared between adjacent cells, and the cells can kind of plug them up. So there's a molecule known as callose. This is a carbohydrate, and it's one that's involved in basically plugging up the connections between adjacent cells. So here it's kind of squeezing that space where there's this connection between two adjacent cells, squeezes it closed. So there's a lot less, as it shows here, a lot less of this flux or flow of molecules in between cells. One place that you see callose plugs is in the growth of pollen tubes. So this allows the pollen, as it grows, we'll see in fertilization, the job of the pollen is to grow a long single strand tube from where the pollen landed to where the ovule is. And along the way, it plugs itself up to kind of mark its progress and to keep the active growing part of that pollen tube at the tip more than anything behind it. I also mentioned how cell walls are not always perfectly rigid. Here's that illustration I was looking for. Cells can expand if their cellulose is oriented in a convenient way for that. And cells also can, if with an existing number of cellulose strands, can determine whether it's stiff or a little more relaxed by these other molecules that hold the cellulose strands together. So that's the job of pectin and hemicellulose, is that they're kind of sticky molecules and they stick the adjacent cellulose strands together. So more pectin and hemicellulose causes it to be stiffer, and less of those molecules would allow things to be more relaxed. And pectin, you might have encountered in this context. Pectin is something you can buy as an additive when you're making jams and jellies. So if you add it to your jelly, it stiffens things up. Some jams, so if you're making something like marmalade, that comes from citrus fruits, and those are pretty good at providing their own pectin. So before people had stores to buy their pectin from, you just add some fruits to your jam that were already rich in pectin, and they would help you with the setting process. But this is kind of a modern cheat we have. We can have just pure pectin and add that and stiffen up things that wouldn't normally be so stiff as jams. And this was their original function, was to hold the cell wall together. So cell wall happens, we often in plants use these terms of primary and secondary, things happening first and second. Plants, when they're initially forming a cell, it gets a primary cell wall, and they invest more or less into this secondary cell wall, which makes the plant cell wall thicker, can have some chemical additives that make it waterproof or more durable. Secondary cell wall is a big part, or a big reason why wood is so stiff. And sometimes you can think about these in terms of what is located where. So we have two cells, the insides of the cells, the cytoplasm of these is in black in this case. And you can see the locations of these different layers as you move from one cell to the other. So the plasma membrane is the farthest toward the inside, as we saw before. The secondary cell wall, if it's present, that's the one that's going to be closer to the cell membrane. It was made second. It was made by the membrane, or by molecules in the membrane. And farther out from that would be the primary cell wall. That was made earlier, was made also by the membrane, but at a location before the secondary cell wall existed. There's this term here, middle lamella, that refers to basically the distance that's where you actually stop being one cell and you start being the other cell. So the middle lamella is the division point between the cell in the upper left here and the cell in the lower right there. And then the same layers exist in the same relative location. So primary cell wall of the second cell, secondary cell wall, plasma membrane there. So I think understanding how those fit in order is important for thinking about how they're made and when they're made. And I might ask you about that later. On the left here, this is a collection of cells that have really thick secondary cell walls. So the primary cell wall is not very thick at all, but if you see a thickening in cells in cross-section, that's due to their secondary cell wall. So illustrating here on the left, plants that don't really have a secondary cell wall, And then a couple of variations on thickenings. Cell walls of xylem cells that conduct water, they are thick. The red on here is a stain that shows up one of the chemicals that typically we find in xylem. So a thick waterproof cell wall there for the water conducting cells. And then over here, this is the pit of a cherry or a plum or something like that, a stone fruit. And you can imagine the hardness of those. I don't know if you've tried to get into one of those before. They're very hard. They call them stone fruits. They're as hard as rocks. And these are cells that are just modified to be hard. So there's like almost no cytoplasm, and they're just all thick secondary cell wall. Another place you might encounter those, the grit, if you eat a pear, those little gritty balls in there are made up of cells that are kind of like this one. So a little bit of a deterrent to animal chewing. Xylem and these other thickenings, one of the molecules that helps them, or really the important molecule that helps them be so thick is known as lignin. This is kind of the active molecule in wood, and it's a rather complicated sort of a phenolic molecule. And it's also durable, very hard for organisms to break down, and also just physically like a stiffening agent for those cell walls. All right, so we'll see those in a little more detail. We'll talk about lignin as we get to xylem. It's one of the topics that'll come up next when we talk about how different plant cells can be different from each other.